Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem in the comment below. So today we will discuss about the Coombs test. So the Coombs test is used for the detection of the uh, hemolytic anemia, and that is also known as the antiglobulin test, is either of two blood tests used in immunohematology. So the immunohematology means the immune system react with the hemoglobin, which that is the red blood cell, and that is known as the study of this is known as the immunohematology. So the direct and indirect method which that is used, and both is used separately for different mechanism for different desire. So the hemolytic anemia is the detection of the direct Coombs test. So here is the person which that is take the blood sample and take the blood serum, for example, and that is we will detect the autoimmune disorder. For example, the hemolytic anemia. Hemolytic anemia means the bursting of the red blood cell due to the autoimmune disorder. So let's begin to understand what is that. So here we will take the blood sample, and this blood sample contain antibody antigen complexes which that are present in the red blood cell. What that means? It means the hemoglobin which that is contain antigen is contain a carbohydrate antigen polysaccharide which that is attached with the antibodies. These antigen are present in the red blood cell. So that is the sample, and here is the red blood cell. This red blood cell is contain the antibody against the antigen which that is present in the red blood cell. So when attachment of this red blood cell with the antibody is known as the antibody antigen complex formation. So this complex formation will burst the red blood cell and inactivate the red blood cell or dysfunction. Why? Because it is collapsed and it is engulfed by the macrophage or other cell neutrophil which that is neutralize this, and it means the red blood cell deficiency in our blood. So our blood will contain a deficient amount of the hemoglobin which that is used for the trapping of oxygen. So the sign and symptom can be the yellow color, as well as another symptom can be the basically less breathing or something because the hemoglobin less. So autoimmune disorder we will discuss in this method. So here we will discuss the autoimmune disorder first to understand what is that and why this hemolytic anemia used to check it out by a Coombs test. So here is the red blood cell which contain a antigen. This antigen is basically not against our body, but this antigen autoimmunetical. Here the T helper cell recognize this antigen by T cell receptor, and antigen piece will recognize by the MHC class 1 molecule with the CD4 receptor. So that means here when the recognition, so the B cell receptor will be receive this antigen and activate to produce memory cell and plasma P cell, which that is attack the red blood cell due to the after the recognition of the antigen are present in the red blood cell. So that is the autoimmune disorder system. So the T regulatory cell which that is regulate and maintain homeostasis and prevention of this autoimmune disorder. So remember about that. So next here we will discuss about just the direct Coombs test first, and second we will discuss about the indirect Coombs test. So the direct Coombs test means it is basically direct antiglobulin test. So the direct antiglobulin test is here is the hemolytic anemia containing blood which that is attached with the antigen. So how to recognize and how to check this is the hemolytic anemia result positive. So let's begin to understand first. So the Coombs reagents used for it. Coombs reagent is basically contain a antibody IgG. So this IgG is used when Coombs test is put into the vial or some media. So it means it will be react with the antibody which that are present already into the red blood cell for the antigen antibody complexes. So the antigen antibody IgG is added in this way, which that is a Coombs reagent preparation in the lab. So here is the basically formation the Coombs test which contain a IgG antibody which that is attached the all antibody region Fc portion of the antibody of the hemoglobin antigen against the hemoglobin antigen. So that is the basically agglutination formation and clump formation will be occur. So the blood clotting will be occur. So this clump formation is due to the attach the IgG with the more than one antibodies of the antigen of red blood cell against the antigen of red blood cell. So you can see in this diagram to understand. So here first of all we will understand about the primary antibodies. So the primary antibodies mean which that is the hemoglobin is bind with the antibodies against the antigen due to the autoimmune disorder of the hemolytic anemia. So that means when we will put that antibodies, it is known as the secondary antibody which that is the IgG. So the IgG basically when secondary antibody will bind, so that means it is the secondary antibody, but it is also use another mechanism to understand. It means the one more amazing thing which that is understanding. I am saying that another Coombs test is also Coombs reagent use IgM. IgM contain a two portion which there is the binding region while the Fc portion. So let's begin to understand first IgM. So what is the IgM containing Coombs test Coombs reagent? This Coombs reagent is look like this. This antibody is the IgM, that is the secondary antibodies also here. So when bind with the antigen antibody complexes of the hemoglobin, so the Fc portion of that antibody is attached with the IgM. So it means the also clotting will be occur, also agglutination will be occur. So that agglutination is too much less prominent as compared to IgM. So we will add the basically here is we can add more secondary antibodies which that is the IgG. So this IgG can enhance the reaction. So let's here we will discuss about the antibody anti-human globin which that is the AHG reagent caused IgG which that coated erythrocytes to agglutinated. So that means the agglutination will be occur due to the IgG, but the IgM is the another way which that is less agglutination. But again we will add the IgG reagent, Coombs reagent, which that is more prominent clotting. So we can understand as well. This is the hemolytic anemia positive. So the Coombs test positive antigen antibody complexes is found and hemolytic anemia is positive. So that means when the clotting will be occur and agglutination formation will be occur or clump formation will be occur, that means the hemolytic anemia is positive. So that person is contain a hemolytic anemia. So on the other hand, the indirect Coombs test. So what that means? So the indirect Coombs test is used for another purpose. For example, the blood donation. During the blood donation, which we use and check the taking of the blood sample before donation of that blood. So the blood carrier and donor of blood which that is used both sample. So let's begin to understand. One is the blood carrier and one is the donor of blood. which that is before the donation of blood we will check the serum contain antibodies. So in this way the blood carrier which contain antibodies, but it will not bind with the antigen of the red blood cell, so that means it is normal. But important thing is that the serum contain antibodies are present in the plasma of the blood. So it means, what that means, to understand about that, when we will take the blood sample from the donor of blood and we will put the blood drops into our put the blood into the basically drop wise, you can alternate path, so the we can put the serum into the blood, so antibody in serum will be occur. So we will take the blood and put into the test tube and check that result. So what will be happen? So first we take the serum contain antibody which that is come from the blood carrier, which that is not bind and not harmful, while on the other hand the antibody in serum of the blood donor of blood, which that is put that blood into the test tube. So we will check the reaction. What is the reaction? So the antibodies antigen complexes are present, antibody antigen complexes which that are bind. So after this binding we can't detect about that antigen binding because it is not agglutination. Agglutination will be not occur, not clump formation, but we will check by the adding the Coombs reagent, IgG antibodies, which that is the Coombs reagents. So these reagents will be add into the binding site of the antibodies, which that is the antibody of the blood carrier, which contain but not bind but are present into the plasma. So it means when IgG will be added into the red blood cell, so due to the Coombs reagent putting into the blood, so that means the agglutination or clump formation will be occur. So we can say that indirect Coombs test is a positive. So it means the donor can give a agglutination or give the attack of their antibodies of their blood. So that means we will not give the blood if that reaction will be occur. So I can't donate people which that is according to the against the antigen antibodies are present in the carrier, while the donor antigen of the red blood cell which that is attacked by the carrier antibodies. So that means it is harmful for that. So the clump formation will be occur and the carrier can be die. So that means both blood group containing person is not able to transfuse the blood. So that is important. But important thing is that blood carrier is also can't donate to the donor. So that means blood transfusion is difficult mechanism to understand by this indirect Coombs test, by the checking of the agglutination, by the adding of the reagent is known as the Coombs reagent. So important thing is that the blood clotting will be occur in the lab. So we can say that the blood donor which can't donate to the carrier because it is against the antigen antibodies are present. Okay, I hope you make sense about that indirect and direct Coombs test. So thanks for watching. Please make sure to subscribe and bye.